Sir Evolution Chapter 12. Asterisk Chapter 12, Memory, Cognition, Sleep and Biological Clock asterisk. Learning can be defined as a neural process that enables both humans and animals to adapt to their environments by using previous experiences to adjust their behavior. A simple example of learning is thus habituation. On the other hand, memory is the neural process by which experience acquired through learning is stored and eventually accessed. Memory involves a complex network of different brain functions located in various brain regions working together to manage information. For this reason, it is more appropriate to define it in terms of memory systems. These have been identified as a consequence of the increased understanding of the neuroanatomical and neurobiological bases of memory. Asterisk 12.1 Type of memory asterisk. Memory consists of two broad categories, short and long-term memory. Short-term memory is also called working memory. Short-term memory reflects the ability to hold a limited amount of information in a very accessible and temporary state. Finally, working memory is not completely distinct from short-term memory. It is a term that is used to refer to memory as it is used to plan and carry out certain behaviors. Long-term memory is a vast storage of knowledge and a record of prior events, being supported by all theoretical views. It includes both declarative and non-declarative types of memory. Long-term memory can be further divided into declarative and non-declarative memory, Figure 104. Declarative memory is knowing what, and non-declarative memory is knowing how. Declarative, explicit memory, it refers to information that is conventionally transmitted or expressed and supports the capacity to recollect facts, semantic, and events, episodic. A fact like Jaipur is the capital of Rajasthan, or an event like a prior vacation to Goa. The formation of declarative memory is dependent on neuronal pathways anatomically connected in the medial temporal lobe and involving the hippocampus, in particular the dentate gyrus, DG, subregion, the subicular complex, and the adjacent pararhinal, entorhinal, and parahippocampal cortices. Non-declarative, implicit memory. It includes the types of memory systems that do not have a conscious component but are nevertheless extremely important. They include the memories for skills and habits, e.g., riding a bike, driving a car, playing badminton or a guitar, a phenomenon called priming, simple forms of associative learning, e.g., classical conditioning / Pavlovian conditioning, and finally simple forms of non-associative learning such as habituation and sensitization. Such form of learning cannot be orally transmitted, thus being expressed through performance rather than recollection. Another specific type of memory that cannot be strictly assigned to any of these classifications is spatial memory. Indeed, spatial memory encompasses aspects of non-declarative, procedural memory, declarative, semantic, and episodic memory, as well as of both short and long-term memory. Figure 104, Type of learning, declarative and non-declarative. Asterisk 22.2 Decoding brain regions involved in memory asterisk. Where learned information or memories stored into the brain in many different parts of the brain or in complete V-brain. Carl Lashley began exploring this problem about 100 years ago by making lesions in the brains of animals such as rats and monkeys. He was searching for evidence of the engram, the group of neurons that serve as the physical representation of memory. First, Lashley, 1950, trained rats to find their way through a maze. Then, he used the tools available at the time, in this case a soldering iron, to create lesions in the rats' brains, specifically in the cerebral cortex. He did this because he was trying to erase the engram, or the original memory trace that the rats had of the maze. Lashley did not find evidence of the engram, and the rats were still able to find their way through the maze, regardless of the size or location of the lesion. Based on his creation of lesions and the animal's reaction, he formulated the equipotentiality hypothesis. If part of one area of the brain involved in memory is damaged, another part of the same area can take over that memory function. Although Lashley's early work did not confirm the existence of the engram, modern psychologists are making progress locating it. Eric Kandel, for example, spent decades working on the synapse, the basic structure of the brain, and its role in controlling the flow of information through neural circuits needed to store memories. Many scientists believe that the entire brain is involved with memory. However, since Lashley's research, other scientists have been able to look more closely at the brain and memory. They have argued that memory is located in specific parts of the brain, and specific neurons can be recognized for their involvement in forming memories. The main parts of the brain involved with memory are the amygdala, the hippocampus, the cerebellum, and the prefrontal cortex, Figure 105. Figure 105, location of the amygdala, the hippocampus, the cerebellum, and the prefrontal cortex. It is now clear that there are a number of different forms or aspects of learning and memory that involve different brain systems. Broadly declarative, explicit, memories for experience involve the hippocampus medial temporal lobe system and non-declarative, implicit, basic associative learning and memory involves the cerebellum, amygdala, and other systems. Asterisk 12.2.1 The hippocampus asterisk. The hippocampus is part of the limbic system and is a bilateral structure located within the medial temporal lobes of the cerebrum in humans. In rodents, it is an elongated, banana-shaped structure with its long axis extending in a C-shaped manner from the midline of the brain near the septal nuclei into the temporal lobe. The hippocampus proper has three subdivisions, CA1, CA2, and C stands for cornuamones. The other regions of the hippocampal formation include the DG, subicular complex, and entorhinal cortex. The hippocampus is organized such that the main excitatory projections follow a unidirectional pathway. Axons from the entorhinal cortex project to superficial layers of the DG via the perforant pathway, which includes medial and lateral subdivisions. These perforant path axons converge on granule cells of the DG, which then send axonal projections called mossy fibers to synapse with pyramidal cells in the CA3. These neurons transmit information via Schaefer collateral axons to pyramidal cells located in area CA1, which in turn send axonal projections to the subiculum and entorhinal cortex. It is now well established that the hippocampus plays an integral role in the consolidation of declarative memory as well as context-dependent and spatial learning processes. A classic example supporting this assertion is that of Henry Gustav Molaison, known only as HM until his death in 2008. HM suffered debilitating seizures for many years before undergoing a bilateral temporal lobe resection that resulted in the destruction of most of his hippocampus. From that time on, HM suffered severe anterograde amnesia, although his memories of events from before the surgery were mostly unaffected and he was able to learn new procedural tasks. He was unable to form new declarative memories and was heavily impaired in spatial learning tasks. Group of researchers also experimented with rats to learn how the hippocampus functions in memory processing. They created lesions in the hippocampi of the rats and found that the rats demonstrated memory impairment on various tasks, such as object recognition and maze running. They concluded that the DG of hippocampus is involved in memory, specifically normal declarative memory as well as spatial memory, when the memory tasks are like recall tests. Another job of the hippocampus is to project information to cortical regions that give memories meaning and connect them with other connected memories. It also plays a part in memory consolidation, the process of transferring new learning into long-term memory. Functional imaging experiments using rats that were involved in spatial tasks has led to the idea of the existence of specialized place cells in the hippocampal DG that fire in response to location cues. It is currently believed that the specific pattern of firing of these cells allows animals to locate their spatial position with a good degree of precision. Asterisk 12.2.2 The amygdala asterisk The main job of the amygdala is to regulate emotions, such as fear, aggression and associated autonomic responses to deal with the situation, altered heart rate, blood pressure, etc. The amygdala plays a part in how memories are stored because storage is influenced by stress hormones. For example, one researcher experimented with rats and the fear response. Using Pavlovian conditioning, a neutral tone was paired with a foot shock to the rats. This produced a fear memory in the rats. After being conditioned, each time they heard the tone, they would freeze, a defense response in rats, indicating a memory for the impending shock. Then the researchers induced cell death in neurons in the lateral amygdala, which is the specific area of the brain responsible for fear memories. They found the fear memory faded, became extinct. Because of its role in processing emotional information, the amygdala is also involved in memory consolidation, the process of transferring new learning into long-term memory. The amygdala seems to facilitate encoding memories at a deeper level when the event is emotionally arousing. Asterisk 12.2.3 The cerebellum and prefrontal cortex asterisk Although the hippocampus seems to be more of a processing area for explicit memories, cerebellum is responsible for implicit memories such as procedural memory, motor learning and classical conditioning. For example, one classical conditioning experiment is to accustom subjects to blink when they are given a puff of air. When researchers damaged the cerebellums of rabbits, they discovered that the rabbits were not able to learn the conditioned eye blink response. Other researchers have used brain scans, including positron emission tomography, PET, scans, to learn how people process and retain information. From these studies, it seems the prefrontal cortex is involved in processing and retaining information. In one study, participants had to complete two different tasks. Either looking for the letter A in words, considered a perceptual task, or categorizing a noun as either living or non-living, considered a semantic task. Participants were then asked which words they had previously seen. Recall was much better for the semantic task than for the perceptual task. According to PET scans, there was much more activation in the left inferior prefrontal cortex in the semantic task. In another study, encoding was associated with left frontal activity, while retrieval of information was associated with the right frontal region. Asterisk 12.2.4 Neurotransmitters asterisk There also appear to be specific neurotransmitters involved with the process of memory, such as epinephrine, dopamine, serotonin, glutamate, and acetylcholine. There continues to be discussion and debate among researchers as to which neurotransmitter plays which specific role. Although we don't yet know which role each neurotransmitter plays in memory, we do know that communication among neurons via neurotransmitters is critical for developing new memories. Repeated activity by neurons leads to increased neurotransmitters in the synapses and more efficient and more synaptic connections. This is how memory consolidation occurs. It is also believed that strong emotions trigger the formation of strong memories, and weaker emotional experiences form weaker memories. This is called arousal theory. For example, strong emotional experiences can trigger the release of neurotransmitters, as well as hormones, which strengthen memory. Therefore, our memory for an emotional event is usually better than our memory for a non-emotional event. When humans and animals are stressed, the brain secretes more of the neurotransmitter glutamate, which helps them remember the stressful event. This is clearly evidenced by what is known as the flashbulb memory phenomenon. A flashbulb memory is an exceptionally clear recollection of an important event. Where were you when you first heard about the 26/11 Mumbai terrorist attacks or when India won the Cricket World Cup in 2011? Most likely you can remember where you were and what you were doing. Thus, we can conclude that three brain areas do play significant roles in the processing and storage of different types of memories: cerebellum, hippocampus, and amygdala. The cerebellum's job is to process procedural memories. The hippocampus is where new memories are encoded. The amygdala helps determine what memories to store, and it plays a part in determining where the memories are stored based on whether we have a strong or weak emotional response to the event. Strong emotional experiences can trigger the release of neurotransmitters, as well as hormones, which strengthen memory. So that memory for an emotional event is usually stronger than memory for a non-emotional event. This is shown by what is known as the flashbulb memory phenomenon, our ability to remember significant life events. 2.3 Neural mechanisms of learning and memory. It is generally accepted that the neurobiological mechanisms underlying the processes of learning and memory formation include various forms of synaptic plasticity involving neuronal networks that are activated during learning. Since the discovery of long-term potentiation, LTP, in 1973, the role of synaptic strengthening in learning and memory has been the subject of considerable investigation, and numerous studies have provided new insights into how this form of plasticity can underlie memory function. There are, however, other forms of synaptic plasticity, including synaptic elimination or weakening through long-term depression, LTD, synaptogenesis, i.e., the growth of new synaptic connections, synapse remodeling, and neurogenesis, i.e., the generation of new neurons in the adult brain, and several lines of evidence have also implicated these additional forms of synaptic plasticity in learning and memory. Importantly, the two functional forms of neuronal plasticity, LTP and LTD, were first discovered to occur in the hippocampus, and the hippocampal DG is one of the two regions in the adult mammalian brain that sustains neurogenesis throughout adulthood, further implicating this brain region in certain types of learning and memory. 12.3.1 Synaptic strengthening and long-term potentiation. LTP refers to a long-lasting increase in the size of an evoked postsynaptic potential or current. The physiological and pharmacological characteristics of LTP make it a viable mechanism for learning and memory, as it can be long-lasting, from weeks to several months, and is input-specific and associative in nature, endowing it with the ability to process converging input in a manner reminiscent of associative learning. Early theories of learning and memory postulated that memories might be formed by strengthening the connections between existing neurons, thereby improving the effectiveness of their communication. In agreement, recent studies have confirmed that increases in synaptic strength occur in different brain regions during the formation of memories. 4. Example, LTP was observed in hippocampal circuits during forms of associative learning that require an intact hippocampus, and in the motor cortex during procedural learning. While several mechanisms of LTP have been elucidated, one of the best characterized is mediated by the glutamate N-methyl-D-aspartate (NMDA) receptor. Importantly, NMDA receptor-dependent LTP exhibits several properties that make it an attractive candidate as a memory mechanism, being suggested to play an incidental role in memory formation rather than in memory storage and retrieval. Furthermore, it has also been postulated to be the neuronal equivalent of an arousal or attention device. LTP induction occurs when repetitive synaptic activation leads to the release of the excitatory neurotransmitter glutamate, and the consequent depolarization of the postsynaptic cell through the activation and opening of glutamatergic amino 3-hydroxy-5-methyl-4-isoxazolepropanoic acid (AMPA) receptor channels. The combined effect of AMPA-mediated depolarization and glutamate binding to postsynaptic NMDA receptors relieves the magnesium blockade of these receptors and allows the entry of calcium ions. The rapid rise in intracellular calcium concentration triggers the activation of protein kinases, including calcium/calmodulin-dependent protein kinase 2, CAMKII, and protein kinase C, PKC. One of the substrates of both CAMKII and PKC is the AMPA receptor, and the phosphorylation of existing AMPA receptors by these Kinases results in an increase in their activity, as well as the insertion of additional AMPA receptors into the postsynaptic membrane, Figure 106. Figure 106, recruitment and opening of AMPA receptor during long-term podotenylization. While early phases of LTP are dependent on protein phosphorylation, maintenance of LTP over a long period of time requires gene transcription and protein synthesis to occur in the postsynaptic cell. The persistent activation of protein kinases, including protein kinase A, pKa, calcium/calmodulin-dependent protein kinase 4, CAMKIV, and mitogen-activated protein kinase, MAPK, leads to the activation of the transcription factor cyclic AMP response element binding protein, CREB, and consequently, to cyclic AMP response element, CRE, mediated gene expression. It is believed that these changes in gene expression may trigger the synthesis of proteins that underlie the maintenance of LTP. Such proteins may contribute to an increase in dendritic spine number, spine surface area, and postsynaptic sensitivity to glutamate. 12.3.2 Synaptic weakening and long-term depression. Synaptic weakening, or the retraction and disappearance of synaptic connections within complex networks, may reflect retrieval failure. Alternatively, it may serve as a means to weaken and use connections, thereby promoting the emergence of patterns of reinforced connections in the network, which could potentially benefit the storage of information. Indeed, synaptic elimination or pruning is known to play a crucial role in specifying the wiring of neuronal circuits during development. Moreover, evidence suggests that these processes can also play an important role in learning and memory. The discovery that activity-dependent LTD, a long-lasting reduction in the efficacy of neuronal synapses, can occur at hippocampal synapses under certain conditions of stimulation indicates that neurons have the mechanisms to reduce synaptic strength in a form that can be functionally equivalent to synaptic elimination or selection. It is thought that LTD can occur at most excitatory synapses in the brain, allowing for bidirectional synaptic plasticity to be established. In the hippocampus, DG and CA subregions, LTD is typically induced by repetitive stimuli at low frequencies, 0.5 to 5 Hz, for extended periods of time, 10 to 15 minutes. The induction of LTD can also require the activation of NMDA receptors. However, a small, slow rise in postsynaptic calcium levels is thought to induce LTD, as opposed to the sharp increase in intracellular calcium concentration that is responsible for inducing LTP. Furthermore, calcium release from intracellular stores has also been proposed to contribute to LTD. While the activation of protein kinases is essential during the early phase of LTP, the activation of calcium-dependent phosphatases such as calcineurin and protein phosphatase 1, PP1, is involved in the early stages of LTD, Figure 106. These protein phosphatases dephosphorylate postsynaptic PKC and pKa, while CAMKII remains relatively unaffected. Furthermore, LTD is also associated with the dephosphorylation of postsynaptic glutamate AMPA receptors, which results in a decrease in the receptor open channel probability and its removal, i.e., internalization, from the cell membrane. It has been suggested that the LTD-mediated synaptic weakening that occurs during memorization of a novel context may result in a greater potential for LTP-mediated synaptic strengthening during the subsequent synaptic recruitment that occurs when animals learn an explicit memory related to that context. 12.3.3 Synaptogenesis and synapse remodeling. Although it has long been speculated that structural rearrangements and remodeling of neuronal networks could be one of the mechanisms underlying learning and memory, both LTP-mediated synaptic plasticity and behavioral learning are accompanied by structural modifications of synaptic connectivity. However, while the formation of new spines and synapses has been demonstrated to occur following LTP induction, there is no direct evidence that the same process also occurs during learning. Nevertheless, both LTP and learning can remodel existing neuronal circuits through a range of morphological changes that can lead to increased synaptic strength and the appearance of new functional synaptic units. Asterisk 12.3.4 Adult neurogenesis asterisk kissing face. During the past 50 years it has become convincingly apparent that the adult mammalian brain retains the capacity to generate new neurons, a characteristic that is preserved in humans. This process is not ubiquitous, however, in neuronal proliferation and differentiation only continue to occur in restricted areas of the brain. Interestingly, the DG of the hippocampus is one of such regions where neurogenesis occurs during adulthood. Here, newborn neurons migrate just a short distance from the DG subgranular zone to the DG granule zone where they integrate into the existing circuitry. A dividing progenitor cell gives rise to daughter cells which differentiate, migrate, and integrate, extending dendrites towards the molecular layer and an axon towards the CA3 region of the hippocampus. Adult neurogenesis and each one of its phases are tightly regulated and can be influenced by many factors. While being partially regulated by genetics, the generation of new neurons in the adult brain is also regulated by physiological, pathological, and behavioral factors. For example, stress, inflammation, and aging can downregulate adult neurogenesis. Conversely, antidepressant drugs, growth factors, physical exercise, environmental enrichment, and learning can upregulate the capacity for neurogenesis in the adult mammalian brain. Importantly, newly generated neurons have particular physiological properties that make them more susceptible to behavioral-dependent synaptic plasticity. Indeed, new DG neurons present a low threshold for LTP induction and the ability to produce stable LTP more readily than MADACE neurons, which might be due to their specific membrane/properties such as greater NMDA receptor sensitivity and calcium entry upon synaptic activation. On the other hand, LTP has also been shown to induce adult hippocampal neurogenesis, which further strengthens the link between structural and functional hippocampal synaptic plasticity. Since these newly generated neurons are linked to the functioning of the hippocampus, it is reasonable to speculate that they might play a role in the mechanisms of hippocampal-dependent learning and memory. In agreement with this hypothesis, numerous correlative studies have shown that hippocampal neurogenesis can be modulated by learning and behavioral experience, and that loss of hippocampal neurogenic function can have consequences on memory formation. In a pioneer study, both spatial learning in the Morris-Water maze and trace eye blink conditioning were shown to enhance the survival of newborn neurons in adult rats, whereas learning tasks that do not involve the hippocampus had no effect on neurogenesis. Similar findings have since then been reported by others. However, various studies have failed to detect an effect of hippocampal-dependent learning on adult hippocampal neurogenesis. The reasons for these discrepancies are not fully understood, but may involve difference in the methods used to induce learning as well as in the protocols utilized to evaluate neurogenesis, e.g., use of different neurogenic markers. Nevertheless, it was recently shown that new neurons are indeed recruited into neuronal circuits involved in spatial learning and memory in the hippocampus. Furthermore, other studies have shown that disrupting or ablating adult hippocampal neurogenesis results in impaired hippocampal-dependent learning and memory. Indeed, experimental reduction of adult neurogenesis impaired hippocampal-dependent trace eye blink conditioning but not hippocampal-independent delay conditioning. Similar results were obtained with other hippocampal-dependent tests, including place recognition tasks, contextual fear conditioning, and a non-matching to sample task, which measured conditional rule learning and memory for specific events. Based on these studies, it is currently believed that hippocampal new neurons are required for the separation of events based on their spatial and temporal characteristics, a process that preserves the uniqueness of a memory representation. as well as space representation, long-term memory retention, and flexible inferential memory expression. 2.4 Animal models for learning and memory. Animal models have provided a rich source of information about the neurobiological basis of learning and memory and have been instrumental in shaping our understanding of how the normal and damaged or diseased brain processes information. 12.4.1 Rodent models of conditioning. Associative learning occurs when an animal learns to connect two events with each other, and tests of conditioning continue to be the most important techniques to study associative learning and memory. Conditioning can be further divided into classical conditioning, a form of associative learning that involves the association between two stimuli, and operant conditioning, which occurs when a response and a stimulus become associated. Rodent models of classical conditioning. The first model of classical conditioning was developed by Ivan Pavlov, who noted that salivation in a dog, normally evoked by a food placed in the dog's mouth, could also be evoked by stimuli that, on repeated occasions, had preceded the feeding of the dog. In light of his pioneering contribution to the field, classical conditioning can also be referred to as Pavlovian conditioning. Fear conditioning. In fear conditioning, an unconditioned stimulus, US, such as a tone or static feature of the training context, i.e., light and smell, is paired with a conditioned stimulus, CS, in this case an aversive stimulus. During the course of learning, the CS acquires the ability to elicit a response, which was originally only evoked by the US. The fear conditioning paradigm has been quite successful as a model to study the cellular and molecular mechanisms underlying learning and memory. Furthermore, this behavioral paradigm has potential relevance as a model for human anxiety disorders. Q plus contextual fear conditioning. It is one variation of fear conditioning that is used in studies of rodent learning and memory. In a typical Q plus contextual fear conditioning experiment, animals are placed in a fear conditioning apparatus for about 2 minutes, followed by a 30-second period when an acoustic CS, tone or preferably white noise, is delivered. During the last 2 seconds of the tone, a mild foot shock, US, is applied to the floor grid of the apparatus. This pairing protocol can be repeated with a brief interval, e.g., 2 minutes, between pairings. When trained in this fashion, the animals learn that the context in which they are trained is a place to be feared, and that the noise or light CS predicts an upcoming foot shock, and thus it should also be feared. These two components of the learning are referred to as contextual and cued fear conditioning, respectively. The brain regions that are thought to be involved in the response to these types of fear conditioning are the amygdala and the hippocampus. Eye blink conditioning. Eye blink conditioning uses the association of a neutral stimulus, such as tone or light CS, paired with a nociceptive US, such as an air puff delivered to the eye or a mild periorbital shock. Representation of the CS results in an eye blink. Conditioned response, CR, in anticipation of the US. Depending on the time between CS onset and US onset, eye blink conditioning can be subdivided into delay eye blink conditioning, when the CS onset precedes the US onset and the two stimuli overlap and co-terminate, and trace eye blink conditioning, when the CS precedes the US and there is a stimulus-free period, called the trace interval, between the CS offset and the US onset. Importantly, while both of these procedures require the involvement of the cerebellum, the trace procedure also requires the hippocampus. Classical conditioning of the eye blink response is typically performed using rabbits as the experimental subjects, although the procedure can also be performed in rats and mice. 2. Rodent models of operant conditioning. The operant conditioning task is one of the most important learning paradigms used in rodents for studying goal-directed behaviors. Edward Thorndike first observed operant conditioning in cats in the so-called puzzle box. Animals locked in the box with food outside the door, would eventually by trial and error, push the right levers to open the box and obtain the food. After only a few trials, cats learn to open the box quickly and efficiently. Accordingly, in paradigms of operant conditioning, a stimulus is paired with a response, for example, an animal that must press a lever to receive food as a reward. The animal thus learns from the consequences of its behavior, changing it accordingly. An operant conditioning chamber, also called Skinner box, permits experimenters to study behavior conditioning training by teaching a subject animal to perform certain actions, e.g., pressing a lever, in response to specific stimuli like a light or sound signal. When the subject correctly performs the behavior, the chamber mechanism delivers food or another reward. In some cases, the mechanism delivers a punishment, e.g., foot shock, for incorrect or missing responses. Thus, with this apparatus, experimenters perform studies in conditioning and training through mechanisms of either positive reinforcement, i.e., reward, negative or reinforcement, i.e., punishment. In rodents, two of the areas involved in learning in operant conditioning task are the medial prefrontal cortex and the hippocampus, and recent studies have shown that a reward-dependent task differentially induces structural plasticity, i.e., cell proliferation, cell survival, astrogliogenesis, neurogenesis, and neuronal maturation in these brain regions. Asterisk 12.4.2 Rodent models of spatial learning and memory. Asterisk. Spatial memory evolved in different species, possibly because it provides information on spatial locations, object configuration, and specific routes that are relevant to the preservation and survival of the species. By means of this memory system, animals can locate food sources while preventing risky situations on the basis of previous experiences. Edward Tolman, 1949, was the first to study spatial behavior in rats. Tolman observed that when hungry rats were put at the entrance of a maze and provided food at the end, the error rate to find food decreased with the number of trials. Since the initial experiments by Tolman, evaluation of hippocampal-based spatial learning and memory has been assessed by numerous behavioral paradigms in rodents. Tasks-based learning. Tasks based on mazes are the most frequently used methods to investigate spatial learning and memory in rodents because they use the natural tendency of the animals to navigate through complex environment based on spatial information. The animal begins at a specific starting position in the maze and then must navigate to a goal position in order to receive a reward, e.g., food or water, which constitute examples of positive reinforcement, or avoid a negative event, e.g., foot shock, bright light, or cold water, which represent examples of negative reinforcement. Although there are different mazes that can be employed to evaluate spatial learning and memory in rodents, those more commonly used are the Morris Water Maze, MWM, the Barnes Circular Maze, the Radial Maze, and the T-Maze. Table 16. Asterisk 1. Morris Water Maze. Asterisk. One of the most widely used procedures to study spatial memory is the MWM. The water maze apparatus, Figure 107, consists of a large, circular pool filled with opaque water in which a small escape platform is hidden, spatial version of the test. The animal has to locate the platform hidden under the water from 4 different starting points, or quadrants. Over a number of trials, the animal learns the location of the hidden platform based on distal cues, and with time the latency to locate the platform decreases. The memory is tested afterwards by a probe trial in which the hidden platform is removed and the amount of time spent in the former region of the platform is measured. Both learning and memory tasks are hippocampal dependent, as hippocampal lesions can compromise the performance in this classical version of the MWM. On the other hand, animals can also be assessed in the MWM for their ability to locate a visible platform independently of its spatial location within the maze environment. Importantly, this non-spatial version of the task is unaffected by Hippocampal damage. Figure 107. The Morris Water Maze, MWM, apparatus. The use of the MWM to assess learning and memory presents several advantages. To begin, learning has been shown to occur faster on the MWM when compared to other mazes, possibly due to the exposure of the animal to water, which can be perceived as an aversive stimulus. Furthermore, and in contrast to the radial maze, this test does not require water or food deprivation, thus limiting the number of days needed to proceed with experimentation and eliminating additional stress related with hunger or thirst. Moreover, the use of water in this maze eliminates the possibility of animals using aromatic cues to orient themselves in the escape search, which is a potential confounding factor that occurs in the dryland mazes. On the other hand, although simple to build and adapt, the MWM requires video recording systems and software for complete analysis of behavioral parameters, equipment that may not be readily available to all researchers. Finally, the exposure of animals to the water might be perceived as a stressful event, which can potentially add an external confounding variable to the study. Asterisk 2. The Barnes Maze asterisk. Carol Barnes developed a circular hole board maze task to use as an assessment of spatial learning in aged rats. The Barnes maze consists of an elevated circular platform with holes along its perimeter, 12, 20, or 40 depending on the diameter. The maze is surrounded by black curtains with visual cues placed on them, figure 108. Rodents find open, well-lit spaces aversive and they will search around the platform trying to find a safe, dark refuge. The escape hole is always placed in the same location of the room with respect to distal cues in the environment, and animals use these visual cues in the extra maze environment to learn the spatial location of the escape hole. During testing, animals receive reinforcement to escape from the open platform surface to a small, dark, recessed chamber located under one of the holes, called the target box. Figure 108, Barnes Maze. The use of the Barnes Maze presents some advantages over the three other most popular tests of visual-based spatial learning and memory, MWM, radial maze, and T-maze. First, in contrast to the radial maze, food or water deprivation are not required to motivate animals in the Barnes maze, and therefore performance is not influenced by individual variability in hunger or thirst. Moreover, walking in the Barnes maze is less stressful and less physically exhaustive than swimming in the MWM, and the performance of animals is not influenced by reduction of body temperature caused by the session of swimming over multiple training trials which are required in the MWM. In addition, like the MWM, it allows for the evaluation of learning, working memory, and spatial reference memory. One disadvantage of the Barnes maze is that learning can be very slow or even absent in some cases. This can be explained by the lack of stressful stimuli, thereby producing more exploratory behavior than escape responses in animals that are not sufficiently motivated to escape. Another disadvantage of the Barnes maze is that it can also stimulate non-spatial strategies. For example, if the maze is not cleaned appropriately, the animals can use aromatic cues to solve the maze. Asterisk radial maze. The radial maze was developed by Olton and Samuelson in 1976 and consists of 8 to 17 equally spaced arms radiating from a central platform, 5 cm wide and 35 cm long for mice, or 10 cm wide and 50 cm long for rats, figure 109, which the rodent has to enter in order to obtain a reward of food or water that is placed in some of the arms. An animal that has been deprived from either food or water is placed in the center and is allowed to collect food pellets or drink water from each arm, sampling without replacement. The optimal strategy for obtaining all the rewards in the least amount of time is to visit each arm only once during a trial. The number of visits to empty arms is calculated as errors made and can give information regarding the animal's spatial working memory. Figure 109. The radial maze apparatus. Alton also developed a variant of the radial maze that allowed assessing reference memory in addition to working memory. To test both, only 3 or 4 arms are baited with a food or water reward. Re-entry into a previously baited, now empty arm is defined as a working memory error. Entries into never baited arms are counted as spatial reference memory errors. Further studies have confirmed that the animal uses spatial cues to recognize the arms previously visited, making this an hippocampal-dependent task. However, while this task permits the examination of both reference and working memory, major limitations include the use of food or water deprivation, as well as the presence of odor confounds. Asterisk 4. T-maze asterisk. T-maze tasks are well characterized and are widely used for cognitive behavioral testing in both mice and rats. The T-maze is an elevated or enclosed apparatus in the form of a T placed horizontally, Figure 110. Figure 110, T-maze. Animals are placed at the base of the T and allowed to choose between the 2 goal arms located at the other end of the stem. If 2 trials are given in quick succession, the natural tendency of rats and mice in a T-maze is to alternate their choice of goal arm. Alternation reflects the motivation of the animal to explore its environment and locate the presence of resources such as food, water, mates, or shelter. Therefore, in some versions of the test, a reward of food or water is placed at the end of the goal arms, and animals are deprived of food or water before being submitted to the task. However, animals do not need to be deprived of such resources to show alternation behavior, as they tend to show spontaneous alternation as a form of exploratory behavior. This strategy involves the use of working memory, since the response obtained in each trial varies according to what the animal has previously just done. Alternation behavior, whether rewarded or spontaneous, is excellent for detecting hippocampal dysfunction reference memory errors. Further studies have confirmed that the animal uses spatial cues to recognize the arms previously visited, making this an hippocampal-dependent task. The original T-mazes required constant handling of the animals, potentially inducing stressful responses that could affect the results of the test. For this reason, an alternative T-maze called continuous alternation task has been developed. In this apparatus, removable doors to the entrance of each arm have been built in order to block the access to the unselected arm. With this simple modification, it was possible to avoid excessive manipulation and thus considerably decrease the procedural stress. Asterisk 12.5 Cognition asterisk. Cognition is broadly defined as the acquisition, storage, retrieval, and use of information for decision-making, and usually is applied to learning that involves more than basic associations. Cognitive ethologists have used many approaches to study the cognitive abilities of animals, including those that focus on tool use, problem-solving, numerical abilities, spatial memory, and self-recognition. Asterisk 12.5.1 Tool use asterisk. The ability to use tools is one aspect of cognition. Many species use tools to acquire food that otherwise would be unavailable. Examples include sticks used by chimpanzees, finches, and crows to probe holes to feed on hidden insects. Stones used by primates to crack open nuts. Dolphins uses sponges to feed on fish hidden in sharp rubble on the ocean floor. Selection can favor tool use when it enhances foraging ability. In many cases, individuals learn to use a tool by trial and error after having observed others. But some species appear capable of understanding the problem to be solved by the tool and then discover a functional solution without trial and error learning. One approach to understand this enhanced cognitive aspect of tool use is to examine how individuals select a tool to solve a problem. For example, bearded capuchin monkeys, Cebus libidinosus, use selected stones to use as hammers to open palm nuts. Typically, a nut is placed on a log and a stone is dropped on the nut to open it. Success in obtaining food requires the use of heavy stones that will not fracture during use. These monkeys use cognitive ability that suggests an understanding of the relevant feature of a functional tool, its weight, independent of its size. Asterisk 12.5.2 Problem solving and insight learning. Asterisk. Insight learning is a spontaneous problem solving without the benefit of trial and error learning. Insight learning is like a lightning of a bulb in mind, and you say, yes, I got a solution to this problem. Wolfgang Kohler is pioneer in study insight learning in chimpanzee. In insight learning, the animal uses cognitive or mental processes to associate experiences and solve problems. The classic example is the work of Wolfgang Kohler, 1887 to 1967, on chimpanzees that were trained to use tools to obtain food rewards. He observed that they obtained bananas suspended from the ceiling and out of reach by first stacking boxes to make a platform, figure 111. They then climbed on top of this structure to reach the bananas. In one experiment, he placed bananas outside of the chimp's enclosure and placed several sticks in the enclosure that they could use to reach the food. A chimp named Sultan quickly figured out how to put two sticks together so that they were long enough to reach the food. In these experiments, the animals had no prior experience with the problem and yet appeared to solve it rather quickly, as one would expect from individuals that possess the capacity for insight learning. Figure 111, Insight learning in chimpanzee. Asterisk 12.5.3 Numerical competency. Asterisk. It is the ability to recognize numerical quantities. Irene Pepperbird demonstrated quite sophisticated numerical competency in an African gray parrot, Pasitacus erythicus, named Alex, that was trained to vocally label objects and their color. Alex could correctly answer questions such as how many blue keys and discriminate numbers up to 9. Numerical competency in wild is studied in New Zealand robins, Patroica australis. The birds live in forests and regularly capture large invertebrates that they dismember into smaller pieces before eating. These food items are frequently cached for later consumption in depressions in tree branches. Birds rely on cached food to survive the winter, but caches may be pilfered by others. It may therefore be important for an individual to assign the cache's relative importance. A cache with many food items should be more valuable than one with a single item. Such an assessment requires numerical competency. Wild New Zealand robins have a sophisticated numerical sense when dealing with a small number of items, and that this ability is not developed through extensive prior training. Some species of birds have larger brains than others. According to cognitive buffer hypothesis, large brains, despite their costs, provide survival benefits through flexible behaviors that are learned. This hypothesis states that large brains increase individuals' capacity to learn and allow them to survive and reproduce better in unpredictable, changing environments. Experimental studies indicate that sleep deprivation leads to cognitive impairments of problem solving and insight learning. Sleep deprivation appears to be most severe with learning and memory processes that require the hippocampus, which suggests that this brain region is particularly sensitive to the consequences of sleep loss. Animals vary in their cognitive ability, the acquisition, storage, and use of information. Studies of cognition have often examined numerical competency, tool use, and the ability to solve problems. Recent work has turned toward seeking links between cognitive ability and fitness and examining whether individuals assess the cognitive abilities of potential mates. Work on animal cognition is particularly intense in cetaceans, primates, and dogs and has led to ethical debates about the treatment of captive animals. This area of research has also led to a discussion of animal consciousness, or whether or not animals think about objects or events. Ongoing work examines animals' neural capacity for consciousness and focuses on behavior that suggests animal self-awareness. *2.6 Neural basis of sleep and arousal*. Sleep appears to be fundamental to animal life, yet little is known about how sleep has evolved throughout the animal kingdom. In diverse animal taxa, poor sleep can have detrimental effects on development, cognitive abilities, and lifespan. And it is now appreciated that normal sleep is fundamental to healthy physiology and bodily function. While the function of sleep remains unknown, studies have identified relationships between sleep and anatomical, physiological, or ecological traits. For example, in mammals, parameters such as diet, brain size, social hierarchy, and body mass index all affect total sleep times. A great deal of variation exists in sleep duration and timing among different animal phyla, with animals such as the African elephant sleeping only 3 to 4 h a day, while many animals spend the majority of their time sleeping, including the armadillo, which sleeps over 18 h per day. Even among humans, sleep times vary widely, ranging from less than 5 h to 10 h or more. Despite a widespread appreciation for the diversity in sleep duration between and within species, surprisingly little is known about the relationship between sleep and an animal's ecological and evolutionary history. Sleep is characterized in different ways, depending on the available methods in the organism of study. The two hallmarks classically have been I. Electrophysiological or 2. Behavioral changes associated with sleep-like states. Importantly, there is a strong correlation between sleep-like states defined by electrophysiological and behavioral criteria in animals, ranging from invertebrates to mammals. Asterisk 12.6.1 Electrophysiological definition of sleep. Electrophysiologically, sleep is characterized by changes in brain activity, wave as measured by the electroencephalogram, EEG, in mammals, or local field potential recordings in invertebrates. The EEG has established its place in sleep research and provides the ability to compartmentalize sleep into 3 different stages based on unique patterns of brain wave activity: I. Waking periods, 2. Non-rapid eye movement, NREM, slow-wave sleep, and 3. REM or paradoxical sleep. The awake state is heterogeneous, characterized by desynchronized EEG oscillations of low amplitude and mixed frequencies, and a variable amount of muscle activity. Active or motivated wakefulness is rich in theta, 4 to 9 Hz, and gamma, 40 to 300 Hz, EEG frequency ranges, whereas quiet wakefulness is characterized by slower EEG frequencies, including alpha, 7 to 15 Hz, and beta, 8 to 30 Hz. Using EEG and EMG recordings, it is possible to recognize 2 distinct sleep states: non-rapid eye movement, NREM, and rapid eye movement, REM, sleep that alternate cyclically across sleep. NREM sleep is characterized by high-amplitude low-frequency delta oscillations, 0.5 to 4 Hz, and spindles, bursts of 7 to 15 Hz. Oscillations, in the EEG, and low postural muscle tone. The EEG during REM sleep is dominated by theta and gamma oscillations, with a complete loss of muscle tone in axial posture muscles, REM muscle atonia. Animals in a sleep-like state, as measured by an EEG, typically assume stereotypical behaviors, and, by carefully correlating behavior with changes in EEG patterns, behavioral identifiers have been established. Nevertheless, a number of limitations to electrophysiological recordings, including difficulty of recording in small animals and impracticality of recording in a natural setting, highlight the need for behavioral observations that can be used to define sleep. Asterisk 12.6.2 Behavioral definition of sleep. Asterisk. Behaviorally, sleep is defined as a period of quiescence with a species-specific body posture and/or sleeping site and an elevated arousal threshold. Behaviorally, sleep can be characterized by 5 criteria: prolonged behavioral quiescence, if which is reversible upon stimulation, to differentiate from torpor or coma; 2. A species-specific posture, increased arousal; threshold to respond to external stimuli, and rebound following sleep deprivation, the establishment of behavioral definitions of sleep has opened the door to investigating sleep in small model systems that are not amenable to EEG analysis. Although EEG measurements are typically restricted to a limited subset of taxonomic groups, behavioral definitions are largely generalizable to all animals in the animal kingdom, and permit the investigation of sleep from a comparative perspective, as well as in small genetically amenable animal models. 12.6.3 Mammalian neuronal circuitry of sleep-wake states. Complex interactions between subcortical neuromodulatory neurons in the brainstem, midbrain, hypothalamus, and basal forebrain, BF, the thalamus, and the cortex drive behavioral, physiological, and electrocortical sleep-wake states. Wake-promoting populations project to various structures through a dorsal and a ventral pathway. The dorsal pathway innervates the thalamus, which facilitates transmission of sensory information to the cortex. The ventral pathway innervates the hypothalamus, BF, and the other forebrain structures, which together excite the cortex. It is thought that wakefulness is achieved when both the dorsal and the ventral pathways are activated. Historically, monoaminergic and cholinergic producing neurons were assigned as key players in the regulation of arousal states, yet recent findings reveal important roles for fast gamma-aminobutyric acid, GABA, and glutamate neurotransmission in sleep-wake regulation. Among the wake-promoting monoaminergic and cholinergic populations are the noradrenergic locus coeruleus, LC, dopaminergic ventral tegmental area, VTA, dopaminergic and serotonergic, 5-HT. Dorsal raphe nucleus, DRN, histaminergic tuberomammillary nucleus, TMN, and cholinergic pedunculopontine and laterodorsal tegmental nuclei, PPT/LDT, and BF neurons, figure 112. Importantly, monoaminergic and cholinergic neurons are essential for learning, motivation, attention, reward, mood, and locomotor behaviors in addition to sleep-wake regulation. Figure 112, schematic of neuronal wake-promoting populations. BF, basal forebrain, DA, dopamine, DR, dorsal raphe, HORT. Hypocretin, histamine, LC, locus coeruleus, LH, lateral hypothalamus, NA, noradrenaline, PB, parabrachial area. PPT/LDT, pedunculopontine and laterodorsal tegmental nuclei, TMN, tuberomammillary nucleus, VTA, ventral tegmental area. Hypocretin neurons. The hypocretins are two alternatively spliced neuropeptides, HCRT1 and HCRT2, produced from a single precursor gene. HORT neurons show high discharge rates during active waking and goal-oriented behaviors, decrease their activity during quiet wakefulness, and are silent during sleep. The coordination between sleep-wake states and the stability of arousal appears to be localized in HCRT neurons. The locus coeruleus *. The LC is the main source of noradrenergic transmission to the cortex. LC neurons fire tonically at 1 to 3 Hz during wakefulness, reduce their activity during NREM sleep, and are nearly silent during REM sleep. The release of noradrenaline in the cortex increases firing rates of pyramidal neurons and electrocortical arousal, while blockade of noradrenergic pathways induces synaptic quiescence. During wakefulness, LC activity seems to be essential for HCRT-induced awakenings. The basal forebrain *. The BF contains 3 major cell groups, cholinergic, GABAergic, and glutamatergic neurons. This brain structure has been traditionally regarded as a wake-promoting region, as its activation induces wakefulness and its lesion results in slow oscillations in the EEG and a coma-like state. Cholinergic neurons of the BF are strongly activated during wakefulness and REM sleep, and their optogenetic stimulation induces cortical activation and transitions from NREM sleep to wakefulness. The activity pattern of GABAergic neurons of the BF is highly heterogeneous. A subset of neurons are maximally active during wakefulness and REM sleep, another subset during NREM sleep, and a minority are maximally active during REM sleep. The activity of BF glutamatergic neurons is also heterogeneous, and their optogenetic stimulation induces sleep-to-wake transitions. Asterisk the ventral tegmental area asterisk. The VTA is a key structure in the regulation of motivational processes and contains dopaminergic, GABAergic, and glutamatergic neurons. Asterisk the dorsal raphe nucleus / ventral periaqueductal gray asterisk. The DRN is a heterogeneous brainstem nucleus that innervates many brain regions implicated in sleep-wake regulation. DRN neurons express various neurotransmitters, including serotonin and dopamine. DRN serotonin neurons were initially hypothesized to play an important role in the initiation and maintenance of NREM sleep, as lesions to the DRN in rats and cats resulted in insomnia. Asterisk the tuberomammillary nucleus asterisk. The TMN of the hypothalamus is the sole source of histamine in the brain. Histaminergic TMN neurons send widespread, yet diffused, projections to various sleep-wake regulatory regions, including to the ventrolateral preoptic nucleus and the basal forebrain. Histaminergic TMN neurons show arousal state-dependent alterations in activity. They fire most rapidly during attentive wakefulness, reduce their activity during quiet wakefulness, and are silent during NREM and REM sleep. Asterisk glutamatergic neurons asterisk. Glutamate is the principal excitatory neurotransmitter in the brain, and global modulation of glutamatergic signaling pathways dose-dependently modulates sleep-wake states. Glutamate levels in the posterior hypothalamus and cortex rise progressively during wakefulness, decline during NREM sleep, and increase rapidly at the onset of REM sleep. Asterisk GABAergic neurons asterisk. GABA has been traditionally viewed as a sleep-promoting neurotransmitter. Most sleep-promoting populations are GABAergic, including those in the preoptic area and parafacial zone. 2.6.4 Neuronal mechanisms regulating sleep-wake states across species. All animals across different phyla sleep. The neuronal, cellular, and molecular pathways associated with sleep are highly conserved across species, suggesting an early and common origin for sleep. In zebrafish, the neurotransmitters and neuromodulators controlling sleep-wake states are located in parallel structures to mammals. For example, in both mammals and zebrafish, HORT and histamine neurons are located in the hypothalamus and noradrenaline in the LC. Genetic and pharmacological manipulations of GABAergic, glutamatergic, monoaminergic, and neuropeptidergic neurons in zebrafish result in similar modifications to sleep and wakefulness as in mammals. In zebrafish, as in mammals, HORT neurons send widespread projections throughout the brain and consolidate wakefulness, whereas ablation of HORT neurons fragments sleep-wake states. Mammals and insects also share similar functions in the neurotransmitters and neuromodulators controlling sleep-wake states. For example, as in mammals, dopamine is a major regulator of sleep-wake states in Drosophila, and enhancement of dopaminergic neurotransmission dramatically reduces sleep and arousal threshold and increases locomotor activity. In addition, noradrenergic and histaminergic activity promotes wakefulness in Drosophila, whereas GABAergic and serotonergic neurotransmission promotes sleep. These similarities, as well as the ease of completing large unbiased screens in simple model systems and the numerous forward and reverse genetic approaches available, have placed these species as prominent models in sleep research. Their study provides important insights into the regulatory processes and functions of sleep. Asterisk 12.6.5 Circadian RHYTM controls sleep-wake cycle. Asterisk cycles of change that complete within 24H are known as circadian rhythms and include examples such as the sleep-wake cycle, body temperature change, and release of hormones such as melatonin and cortisol. In mammals, circadian rhythm is coordinated by the central pacemaker located in the suprachiasmatic nucleus (SCN) of the anterior hypothalamus. It has been hypothesized that the major output region of the SCN, in regard to sleep-wake regulation, is the dorsomedial nucleus of the hypothalamus via the subparaventricular zone, which heavily innervates sleep and wake-promoting nuclei. The central circadian clock is comprised of cell-autonomous pacemaker neurons. The pacemaker neurons contain a molecular transcription translation-based autoregulatory feedback loop in which circadian clock genes rhythmically regulate their own expression as well as the expression of thousands of other genes. Since the clock regulates the timing of the sleep-wake cycle, it is not surprising that some core clock genes are also required for normal sleep timing and duration. For example, knockout of clock or PACE2 causes a reduction in sleep, and double knockouts of the two Cry homologs Cry1 and Cry2 cause an increase in sleep. The downstream transcriptional targets of these genes that are ultimately responsible for effects on arousal have yet to be identified. Biological clock *. Among the important proximate regulators of behavior are those that control its organization through time. A biological clock is any recurrent, endogenous cycle, behavioral or physiological, that persists in the absence of geophysical or environmental temporal cues. Rhythmic behavior is characterized by a series of repeating units or cycles, the duration of which constitutes the rhythm's period, figure 113. The degree of change in response between the peak and trough of the cycle is the amplitude, while any particular part of the cycle is generally referred to as a phase. Figure 113, the basic characteristics of rhythmic behavior. A wide variety of biological rhythms have been identified in both vertebrate and invertebrate species, and these rhythms are typically categorized by the length of a single cycle. 12.7.1 Circadian rhythms *. Our planet turns on its axis once every 24 hours, creating a cycle of environmental conditions that has existed throughout the evolution of life. Daily biological cycles are characteristic of almost all organisms. What is surprising, however, is that this daily rhythmicity does not depend on the 24-hour cycle of light and dark. If animals are kept in constant darkness, at a constant temperature with food and water available all the time, they still demonstrate daily cycles of activities such as sleeping, eating, drinking, and just about anything else that can be measured. The persistence of these daily cycles and the absence of environmental time cues suggests that animals have an endogenous, internal clock. Without time cues from the environment, however, these daily cycles are not exactly 24 hours long. They are therefore called circadian rhythms, from the Latin circa, about, and dies, day. A biological rhythm can be thought of as a series of cycles, and the length of one of those cycles is the period of the rhythm. Any point on the cycle is a phase of that cycle, hence, when two rhythms completely match, they are in phase, and if a rhythm is shifted, as in the resetting of a clock, it is phase advanced or phase delayed. Since the period of a circadian rhythm is not exactly 24 hours, it must be phase advanced or phase delayed every day to remain in phase with the daily cycle of the environment. In other words, the rhythm has to be entrained to the cycle of light and dark in the environment. *1. Entrainment *. The resetting of the circadian rhythm by environmental cues is called entrainment. An animal kept in constant conditions will not be entrained to the 24-hour cycle of the environment, and its circadian clock will run according to its natural period. It will be free-running. It will be slightly out of phase with the cue to which they were originally entrained. Figure 114. Thus the flying squirrel in Figure 114, maintained under constant light and temperature conditions in the laboratory show a systematic drift in their normal circadian activity and circannual testicular and molt cycles respectively. This drift is the reason biological rhythms are given the prefix circa, from the Latin for around or about. The approximate free-running periodicity of many circadian clocks studied follow Askoff's rule, which states that the rate and direction of drift away from 24H depends on light intensity and whether the animal is nocturnal or diurnal in its pattern of activity. Thus in the nocturnal flying squirrel kept in constant darkness, for example, the free-running period of activity starts slightly earlier each night, while in male crickets, Teleogryllus commodus, kept in constant light, normally nocturnal, singing behavior starts slightly later. Figure 114, a free-running circadian rhythm of a flying squirrel under conditions of constant darkness. Animals with free-running circadian rhythms can be used in experiments to identify and investigate the characteristics of stimuli that phase shift or entrain the circadian clock. Under natural conditions, environmental time cues, such as the onset of light or dark, entrain the free-running rhythm to the 24-hour cycle of the real world. In the laboratory, it is possible to entrain the circadian rhythms of free-running animals with short pulses of light or dark administered every 24 hours. A Zeitgeber is any external or environmental cue that entrains or synchronizes an organism's biological rhythms to the Earth's 24-hour light/dark cycle. When you fly across several time zones, your circadian rhythm is out of phase with the real world at your destination. The result is jet lag. Gradually your endogenous clock synchronizes itself with the real world as it is re-entrained by environmental cues. But because your endogenous rhythm cannot be shifted by more than 30 to 60 minutes each day, it takes several days to re-entrain your clock to real time in your new location. This period of re-entrainment is the time during which you experience jet lag, because your endogenous rhythm is waking you up, making you sleepy, initiating activities in your digestive tract, and stimulating many other physiological functions at inappropriate times of the day. Asterisk 2. The circadian clock asterisk. Asterisk clock regulation in invertebrates asterisk. The circadian system in birds is based on three interacting clocks: the eyes, pineal gland and suprachiasmatic nucleus, SCN, of the hypothalamus, the relative importance of each differing among species. Transplant experiments, in which the pineals of sparrows entrained to various 24H light-dark cycles are surgically implanted into arrhythmic pinealectomized birds, have shown that the pineal is the circadian pacemaker in these species. Transplant recipients showing the circadian rhythmicity of the donors. However, in some other bird species, such as the Japanese quail, Coturnix japonica, the pineal is less important in this respect than the eyes and SCN. Blinding, but not pinealectomy, abolishes rhythmicity in quail, as it does in pigeons. Columba livia, but has no such effect in sparrows or chickens. While the eyes undoubtedly provide light information to the clock mechanism, however, entrainment to light cycles in some species appears to be directly via photoreceptors in the brain, a route demonstrated by manipulating the opacity of the scalp and roof of the skull. Between them, these clocks control others in the body through the rhythmic secretion of the hormone melatonin by the pineal gland. Interaction between the three clocks is mediated by neural connections and melatonin secretion. The SCN communicates with the pineal via nerve connections and appears to receive feedback hormonally via melatonin receptors. Clock mechanisms among mammals differ in two important ways from those of birds. First, entrainment to light cycles in mammals does not appear to operate via extraretinal photoreceptors as it does in birds. And, second, the master clock influences the rest of the system through nerve connections rather than hormones. Circadian clock mechanisms have been particularly well studied in rodents. In mammals, the master circadian clock is located in two tiny groups of cells just above the optic chiasma, the place where the two optic nerves cross. These structures are called the suprachiasmatic nuclei, SCN. If a mammal's SCN are destroyed, it loses circadian rhythmicity. Under constant conditions, it is equally likely to be active or asleep at any time of day. Remarkable experiments have shown that circadian rhythms of rest and activity can be restored in an animal whose SCN have been destroyed if it receives a transplant of those nuclei from another animal. Because the restored rhythm has the period of the donor animal, the transplanted tissue clearly controls the recipient's behavior. Circadian rhythms are found in every animal group, as well as in protists, plants, and fungi, but only vertebrates have SCN. Thus, natural selection has produced a variety of circadian clocks. Asterisk clock regulation in invertebrates asterisk in cockroaches, Leucophina maederae and Paraplaneta americana, the circadian clock appears to be regulated by large ocellular fibers extending from cells in the optic lobes of the brain to neurosecretory cells in the thoracic nerves. In fact, cockroaches have two regulatory clocks, since each of the two optic lobes keeps time. Independently in some other insects, such as certain species of moth, brain transplant experiments have shown that communication between the cerebral clock and the mechanism controlling rhythmic activity is hormonal rather than neural. Both hormones and neural connections are important in crustacean clocks, with neurons in the eye stalk regulating the basic activity rhythm and hormones from the X organ, a group of neurosecretory cells in the optic ganglia, determining its persistence. In the mollusk bulla, for example, the cells driving circadian rhythms are in the eyes. In some mollusks, seasonal rhythmicity is associated with changes in the ionic status of the hemolymph and nerve cells. After hibernation, the resting potential of neurons is lowered from 60 MV to around 40 to 50 MV, probably through increased permeability of the membrane to sodium and calcium. This brings the membrane potential nearer to the impulse threshold and may even elicit autorhythmic spiking. This galvanizing effect on neural activity is reversed in the summer when activity levels drop and animals prepare to estivate. Thus the seasonal cycle of activity and torpor in these species appears to be regulated by the chemical environment of the nervous system. Asterisk 12.7.2 Molecular biology of circadian rhythm asterisk. Enormous progress has been made in recent years toward discovering the molecular and biochemical basis of circadian rhythms. In all organisms that have been studied, self-regulating positive and negative feedback loops of DNA transcription and mRNA translation interact to generate circadian rhythms. The main set of genes involved in these loops is homologous over a wide range of organisms, from bread molds to humans. This surprising discovery means that circadian clocks probably arose very early in the history of life on Earth. Many of the genetic mechanisms of mammalian circadian rhythms have been revealed by studying gene mutations that alter circadian rhythms. The story began with a gene called period, PER, that was discovered in fruit flies. Mutations of this gene cause flies to have either short or long free-running circadian periods. Mutations of another circadian gene, timeless, TIM, cause a loss of circadian rhythms in fruit flies. The presence of mRNA for PER and TIM shows a circadian cycle of expression, and with a slight delay, so do the PR and TIM proteins. Thus, the transcription and translation of these two genes have a circadian rhythm, figure 115. But what controls the rhythm? The PR and TIM proteins dimerize in the cytoplasm, and the resulting heterodimer enters the nucleus, where it inhibits transcription of the PER and TIM genes. The PR/TIM heterodimer does this by blocking a heterodimer of two other clock genes, clock, CLK, and cycle, psych, that promotes PER and TIM transcription. This molecular negative feedback loop is just one of several such loops in the circadian clock of a fruit fly, but it is the basic model for a clock mechanism, genes whose products shut down their own expression with a delay. Figure 115, the genetic mechanisms of Drosophila circadian rhythms. The circuit design of the mammalian clock mechanism is similar to that of the fruit flies, but it involves different genes. In mammals, the circadian molecular clock also comprises a group of genes, clock genes, that regulate their own transcription and translation in a cycle that takes approximately 24h to complete, figure 116. This regulation is carried out through a series of interlocking negative feedback loops. The transcription factors BMAL1 and clock heterodimerize and promote the expression of the period, PER1/PER2, and cryptochrome, CRY1/CRY2, genes, the nuclear receptors retinoid-related orphan receptor, ROR, and REVR-BA, and a number of downstream genes referred to as clock-controlled genes, CCGs, including monoamine oxidase A, MAO-A, excitatory amino acid transporter, EAT, and estrogen receptor beta, ERB. In turn, the protein products of the PER and CRY genes dimerize and inhibit the transcriptional activity of clock BMAL1. The precise timing of this process is regulated by several kinases, such as casein kinase 1 epsilon/delta, CK1 epsilon/delta, which regulate the post-transcriptional activity of PR-CRY dimers. RORA and REVR-BA also regulate the transcription of BMAL1, whereby RORA promotes its expression, whereas REVR-BA inhibits it. SIRT1, SIRT1, is believed to regulate this feedback loop at several levels, including through promoting RORA-mediated amplification of MEL expression and acetylation of MEL. Figure 116, Mammalian circadian molecular clock genes feedback loops. Asterisk 12.7.3 Other biological RHTHMS asterisk. Several environmental influences on behavior are rhythmic in nature. We might therefore expect organisms to have adapted to these and entrain their patterns of behavior accordingly. All biological rhythms need not be 24h in length. Some other biological rhythms are ultradian rhythms they are rhythms that occur more frequently than 24h. For example, many hormones, e.g., cortisol, and enzymatic reactions display rhythms only a few hours in length. Infradian rhythms they are rhythms longer than 24h but shorter than 1 year. Cycles of reproductive endocrine activity, such as menstrual cycles in humans and estrous cycles in non-human animals, are examples of this kind of rhythm. Circadial and circumcysigic rhythms Any organism living in the intertidal zone of the seashore is alternately submerged by water and exposed to air. Given the number of environmental factors affected by these cyclical inundations and exposures, it is not surprising that organisms show some associated behavioral rhythms. The circotidal vertical migration cycles of sand-dwelling platyhelminths, polychaetes and diatoms, and cycles of contraction in anemones, filtration in bivalves, and swimming activity in fish are good examples. Some seashore organisms, such as the Periwinkle litorina rudis and some crabs, show semilunar or circumcysigic activity rhythms synchronized to the fortnightly cycles of spring and neap tides. Circulunar rhythms in some animals, behavior is entrained to the 29-day lunar month, thus showing circulunar or circasynotic rhythmicity. The Mediterranean polychaete Platanarius demerlii transforms into its sexual phase and swarms at the water surface in synchrony with the full moon. A similar endogenous rhythm underlies the famous reproductive swarming of palolo worms, Unis viridis, in the Pacific. Among terrestrial organisms, antlion, Myrmelion obscurus, show patterns of pit-building activity that peak at around the full moon. Circannual rhythms in addition to turning on its axis every 24 hours, our planet revolves around the sun once every 365 days. Because Earth is tilted on its axis, its revolution around the sun results in seasonal changes in day length at all locations except the equator. These changes secondarily create seasonal changes in temperature, rainfall, and other variables. Because the behavior of animals must adapt to these seasonal changes, animals must be able to anticipate the seasons and adjust their behavior accordingly. Most animals, for example, reproduce more successfully if they do not produce young in the winter. For many species, change in day length or photoperiod is a reliable indicator of seasonal changes to come. If day length has a direct effect on the physiology and behavior of a species, that species is said to be photoperiodic. If male deer, for example, are held in captivity and subjected to 2 cycles of change in day length in 1 year, they will grow and shed their antlers twice during that year. For some animals, change in day length is not a reliable seasonal cue. Hibernators spend long months in dark burrows underground, away from any indicators of day length, but have to be physiologically prepared to breed almost as soon as they emerge in the spring. A bird overwintering in the tropics cannot use changes in photoperiod as a cue to time its migration north to the breeding grounds. Some species of birds and rodents display yearly fluctuations in gonadal mass and changes in reproductive hormones, e.g., testosterone, estrogen, and these fluctuations persist when the animals are maintained within constant conditions of the laboratory. Hibernators and equatorial migrants have endogenous annual rhythms, called circannual rhythms. Circannual rhythms commonly involve yearly cycles in breeding activity and reproductive endocrinology. In other words, their nervous systems have a built-in calendar, just as circadian rhythms are not exactly 24 hours long. Circannual rhythms are not exactly 365 days long, but usually shorter. Closely related to circannual rhythms are seasonal rhythms, which also approximate 1 year. However, unlike true circannual rhythms, seasonal rhythms are generated in response to environmental cues and do not persist in the absence of these cues.